Bismillah ar-Rahman ar-Rahim. Alhamdulillah wa salatu wa salam ala Rasulillah. Welcome everyone to our event tonight. Alhamdulillah, looking forward to tonight's event. We're just going to give a few minutes for people to join, inshallah. But Jazakumullah khair for attending today's very crucial and important event on the COVID-19 vaccine. And we have two wonderful guests, our local Dr. Hassan Aijazi, and he is a local doctor here at the Swedish Medical Center, and I'll let him introduce himself. And he also invited his brother here, Muaz, who's a chief resident in Pittsburgh. And so they wanted to talk a little bit about the vaccine. I know people have had a lot of questions about the distribution, about the safety, about all kinds of things that people have been emailing in. So I'm looking forward to today's conversation, inshallah. So we'll try to finish up before 9 p.m., inshallah. That is the goal. And if you do have questions, then we can take them during the program if it's relevant. And we'll also have a Q&A session at the end. So if you would like to save your question for the end, then you can wait till then. Or if you feel like it's relevant to whatever is being discussed, feel free to ask in the chat. And I'll be here. Muad is also here to help to make sure the question gets answered, inshallah. All right. So just giving another 30 seconds or so for people to join, and then we'll get started, inshallah. Bismillah. So this is actually a part of our community conversations, which we've been having since the summer. Alhamdulillah, we've had some really excellent topics from people in our community, from the greater Seattle area. They talk about topics that are important to them, that they are experts in. We've had topics on parenting teenagers, on civic engagement, even on Bitcoin by different community members. And so even on Quran memorization, so all kinds of things. We've continued to have these since the summer on at least twice a month, alhamdulillah. So if you'd like to continue to get news about these latest events that we have, then please join the MAPS newsletter on the MAPS website, mapsredmond.org, where you can find out more. We'll be taking a little break for the holidays, and then we'll be back. We have some exciting programs around entrepreneurship and Ivy League colleges. I'm looking forward to those programs, inshallah, in the new year. So with that said, I'm going to go ahead and let Dr. Hassan talk a little bit about his history and his introduce himself, and then we'll get started. Jazakumullah khair. So Hassan, take it away.
Assalamu alaikum, everyone. For those of you who don't know me, my name is Hassan. I am an anesthesia and ICU doctor at Swedish. I've been here for 2 years now, and I'm mostly at Swedish downtown, the First Hill and Cherry Hill campuses. I am not an expert on vaccination. I'm not a microbiologist or immunologist. Those are the people who would particularly be experts. I kind of know what I need to know to help make decisions. And like all of you, I'm sure, have just been curious about the whole thing and did some more reading and research on it. So I'm going to try to share with you kind of what I know. And if there's any questions that you guys have that I do not know the answer to, I'm happy to try to look up the answers to those questions for you and try to get back to you on them. And joining us is my brother Muaz. Muaz is in Pittsburgh. He's a chief resident in internal medicine, and he's at the University of Pittsburgh Medical Center, and they're actually a pretty big medical center. They were working on developing their own vaccine for a while, and so he's pretty well informed in kind of what's been going on as well. So we're just going to try to get through together some of the common questions and concerns people may have, and just some kind of general information. And like Sheikh Adam said, if you guys have any specific questions, feel free to throw it in the chat, and we're happy to kind of do a discussion. We're going to try to keep this for under half an hour for sure, and then just kind of make it open up to discussion. So anything that you guys have, we can address at that point too. All right, Muaz, thanks for joining us. So we're going to kind of jump right into it. We're just going to do a kind of a brief overview, kind of about viruses and vaccines in the first place, just for a couple minutes. All right. So viruses are these like super annoying, tiny little particles, about 1/100th the size of a normal cell. Okay. There's a really big debate in scientific circles about whether they're actually alive or not. They're so small, they can't grow on their own, they can't replicate on their own outside of living inside of a host like a human or another animal or something like that. And they're kind of prevalent all around. And certain ones, when they exist in people or in animals, they can kind of make us sick. Well, do you want to talk a little bit about the different types of viruses?
Yeah, absolutely. Thanks for having me. Two big kind of groups of viruses that we have encountered are DNA and RNA viruses. There's certain, different examples that fall under different categories. But the reason that viruses are important and the reason that we have been talking about viruses basically since the advent, since we've understood them, is there is no directed therapy as such to a virus, right? You have, for a bacterial infection, we can give an antibiotic and that would take care of the bacteria and that can kind of get you on the way to recovery. But for viruses, if you are infected with a virus, you kind of just, the virus will kind of take control of your cell's machinery and start causing harm to your own body. And that kind of, the therapy for that is essentially just what we call conservative management, which means waiting for the virus to kind of wear itself out, and then you are back to your normal state of health. And that's kind of what we encounter with the influenza, if you have the flu or the common cold, for example.
Right. The antivirals are definitely behind what antibacterials or antibiotics are. Antivirals like Tamiflu, like you can take for the flu, not nearly as effective as an antibiotic for a bacterial infection. And so when these little viruses get inside our cells, they take over, they use our own cells' machinery to make proteins. And that's the only way that they can replicate. Like Moz was alluding to, they have a virus as kind of a little genetic blueprint surrounded by a little bit of a capsid. And then they have different features to them. Some have an envelope around them, some don't. And just because of what features they have, they can infect different types of cells, different types of animals, and cause a constellation of different types of symptoms. So the one that you guys that we're dealing with in particular is from the family of viruses called a coronavirus. And viruses are it identifies different types. They're not even called different species because people don't really consider them to be alive. They're just kind of little particles. So what we're dealing with is a coronavirus, like you guys know. Moz going to talk a little bit about our immune system and how our immune system, what that is and how that reacts to viruses.
Yeah, absolutely. Our immune system is actually extremely sophisticated. It's an extremely kind of broad and interesting topic. The way that we've understood our immune system to this point as our research kind of develops is we have extremely specific responses to specific antigens. And what is an antigen? An antigen essentially is just something foreign that comes into your body. It could be a virus, it could be a bacteria. When it comes into your body's immune system says, hey, this thing is foreign and it could potentially be harmful. So we have to figure out how to take care of it, how to neutralize this threat.
Right. So you can consider your immune system to be like our body's own intrinsic army to fight against foreign invaders. Okay, and just
There's
like
a question here, Hassan. There's a question
Yeah.
here. Can you explain what is RNA and DNA?
Moz, you want to take that one?
Yeah, absolutely. So DNA was basically it's a small little kind of two chains of I don't even know how to describe the size of it, but it's very, very tiny and it kind of lives in the nucleus of your cell. And what that does is it makes the blueprint for what's to be created from the cell. Now, think of it as a 3D printer, for example. Your cell is a 3D printer, and you feed in the paper blueprint into the 3D printer, and the 3D printer will print out proteins. And then we kind of use those to navigate therapies. So insulin, for example, is a protein that we give to someone. So you have a high blood sugar, we give you the protein, the ready-made protein insulin, and that takes care of your blood sugar. DNA will make RNA and RNA makes a protein. So think of the DNA and the RNA combination as your 3D printer mechanism. That's kind of how I look at, a simplified an explanation of what DNA and RNA is. DNA is that blueprint that makes the proteins that kind of float around in our system and do different functions.
Yeah, and they're both they can be used by different organisms to contain genetic information. So that's what's going to say, oh, this person's a human, or this thing's a chimpanzee. This thing is just based on what that actual genetic information is. Now in humans, like Moz was saying, DNA is double-stranded, so there's two kind of fibers intertwined, and that has all of these codes that tells us what is the blueprint for what is this cell is gonna become, what color someone's eyes are gonna be, what color their hair is gonna be, et cetera, et cetera, et cetera. Some viruses don't exist with DNA. They only have
So the
RNA.
viruses, they ruin the DNA, or they corrupt the DNA, they corrupt the blueprint, basically.
The viruses themselves usually don't attack the DNA, but our DNA can sometimes be corrupted, and things like cancer. Oftentimes what happens is that you have a fault in your DNA, which changes the genetic coding of your cell, which makes the cell not realize that it's supposed to stop dividing, continuously divides. But the virus itself usually does not mess with our DNA in most types of viruses. I think there's a few more rare types of viruses that could do that. I think HPV, human papilloma virus, might be one of the ones that do that. But what it does is, so what our DNA does is our DNA lives in the nucleus of our cells, right? And what the DNA does, it has something that it says it wants to make this protein. So I want this cell to become a red blood cell. Our DNA is deciding that, okay? When it wants it to be a red blood cell, there's certain things that our red blood cell has that is different from other cells, like it has hemoglobin and things like that. So oftentimes with distinguishing things are called proteins. Proteins are kind of like the bricks to your house. So you're saying, okay, we want the red bricks, or we want, you know, the white bricks that are made of different things. This has different strengths than this. So in order to make those proteins, your DNA is a blueprint, okay? That blueprint, I think using that 3D printer analogy, is gonna make messenger RNA, which kind of takes your stuff outside of the nucleus and then creates like RNA. Then there's something called transfer RNA. And overall, this process is called translation and transcription, but that converts into a protein, okay? Your virus, what it does, your virus oftentimes will come in, it has its own little genetic material, which is not nearly as sophisticated as ours, either DNA or RNA. And just with that little bit of information, all that says is, okay, how do I make my own, how do I replicate myself? How do I make another virus that's just like me? So it continues its kind of life cycle, okay? What it'll do is it'll come into our cell, it'll use our ribosomes, which is what creates the protein, and then using our ribosomes, it'll implant its own genetic material to say make more virus. And then your cell just becomes captured by the virus to just continuously make more and more virus, okay? And I use the word mRNA, messenger RNA. That's something that we'll talk about kind of as we talk about different types of vaccines. Hopefully that answers the question. And we got a little bit really into RNA and DNA, but it's kind of a very technical.
Yeah, exactly. Essentially, this is the blueprint that will feed into your body's way of making proteins that, like Dr. Hassan said, is going to build, you know, the building block essentially for our genetic being. Is this a blueprint that our body uses to build and differentiate itself?
Okay, so just going real quick back to the immune system. So I like the analogy of saying it's kind of like our internal army. It's a pretty sophisticated system. So there's things like cytotoxic T cells, B cells, antibodies, natural killer cells, kind of similar to like you would say like in an army, there's like an archer, there's cavalry, someone's got, you know, sword, someone has an axe, there's spies, there's different ways to kind of confront different enemies. Similarly, our body has a pretty sophisticated system to kind of detect and fight against outside intruders, okay? What vaccinations do is try to figure out a way to teach our body to fight against a specific intruder without having that intruder enter into us first and cause the disease, okay? Traditionally, the different types of the different types of vaccines have things like a live attenuated vaccine, which is where they get something like measles, mumps, and rubella. Those are live attenuated vaccines. They actually get the virus that causes it and they weaken it in a way that it's not going to cause severe disease. And then they give that to you in your arm. And what happens, your body's immune system, then it's kind of like doing a training exercise, right? It'll say, that has something called the virus has something called an antigen. An antigen is a distinguishing characteristic of that virus that makes it different from all of your cells. So then our body's immune system will recognize it. For example, if our uniform is blue, the virus will be wearing red. So then we'll know, oh yeah, that's a virus. Or, you know, some viruses are pretty sophisticated, they're pretty stealthy, but our body has to learn that, you know, this is a foreign intruder, and what are the weaknesses of this intruder? How do I attack against it? Okay. So traditionally, like I was saying, we've used live attenuated vaccines, which is a weakened version of that vaccine. We've used inactivated vaccines. So it's a version of that virus, which is kind of killed, so it can't cause the infection, but it's still introduced to you so that your body can still detect it and figure out how to fight against it. What our body then does is create special, it's kind of like making a wanted poster inside our little city saying that, hey, hey, this is what this guy looks like. Next time you see him, he's not welcome here. So take care of business. And then our body, our immune system creates antibodies, which are specifically detected against that virus or that antigen. It's the exact shape of what that virus is going to be like to prevent it from being able to do its job. Is that right about vaccines, Waz?
Yeah, exactly. You're exactly right. I think what's really important to understand, though, is that when it comes to viruses, your body really requires a very specific response. So each response will be different for each virus that comes in, and your body needs a very specific response to a specific virus. And that's exactly why vaccination is so important when it comes to viruses. Your body needs to know the specific response that is required to take care of that exact virus. And I think that's something that's very important to kind of keep in our minds, kind of going as we talk about vaccines going forward.
Yeah, so for example, within the coronavirus family, there's different types of coronaviruses. So you could be immune to one, either through vaccination or previously being infected with that virus, but still not have immunity to the other coronaviruses because there's a slight difference between them. And you need to have immunity to the new type of coronavirus. Okay, so you want to talk a little bit about, so we talked about the principle of vaccines and the immune system. So let's talk about the new vaccines.
Yeah, absolutely. I think the question that I've kind of come across and the question that I inevitably have wondered myself is, how did we get here so fast? Right? Generally, if you think of a vaccine, you're thinking along the terms of 4, 5 to maybe even 10 years before we've come to an FDA approved vaccine. But here we are 8, 9 months later, sitting on FDA approved vaccinations that we've already started receiving, as far as early as yesterday or into today.
Right.
Sorry, go ahead.
Yeah, I think that's one of the people's main questions is what corners were cut, because we know something is up, and is it safe or not? Like, is it safe to take or not? Moaz, you want to talk about kind of how do vaccines come about in the first place? Like, what is the process of creating a vaccine?
Certainly, yeah. So vaccines, regardless of how fast the process is, vaccines go through a very rigorous process before they are, FDA approved. I'm sure we've been hearing the term FDA approval a lot, in the past month or so. The very first step to it would be an animal trial. We use animal trials to kind of see. So there's two main domains that we use clinical trials for. Number one would be safety. Is the vaccine safe to give? Two, is it effective?
What's a clinical trial? You want to start with what a clinical trial is?
Yeah, absolutely. A clinical trial essentially is looking at, is this therapy worth giving? That's the question that we want, we want answered by a trial. Is something that we're giving, is this either better than something that we already have, or is this something that will be good going forward?
And it's the gold standard of seeing if something is safe and effective.
Right. Yeah, exactly. So safe and effective. Those are the two things that we're looking for. And the process of that kind of runs through, initially we have animal trials, and animal trials kind of fall in the domain of safety. Once we look at animal trials, we kind of start moving towards, hey, this vaccine this therapy is safe. Now we can move on to what we call the human trials, where we go in from the phase 1, 2, and 3. And phase 1 essentially just entails a very small number of people. So it can be 10 to 50, as high as 100 maybe. And that again is looking at safety. And this is looking at safety in terms specifically for dosage. Right? Are we giving too low of a dose to the point where you're not making an immune response, versus are we giving too high of a dose to the point where you're having extreme side effects, right?
Right.
So once we kind of adjust that a little bit, we move on to phase 2. Phase 2 just involves more and more people. And once you've reached phase 2, that's when you really start thinking, hey, maybe this therapy will work. So you start to enroll more and more people, you start to fine tune the dosage, and you start to move away from safety, and you go more into efficacy. Once you've reached phase 3 status, phase 3 is where you start to involve your thousands and thousands of people. I think Pfizer had about 40,000 people in its phase 3 trial. And when you do a phase 3 trial, you want to make sure that you have a pretty wide variety of demographic population that are represented in your trial to make sure that it's accurately represents a real life scenario. So you have your whole demographics, you make sure that's kind of controlled for. But essentially what the phase 3 trial tries to prove is that this trial, this therapy is proven to be safe by the prior phases. Now we're trying to say, is this effective or not? And that's kind of where we are now. We've completed the phase 3 trials, and in Pfizer BioNTech's terminology, we've completed the phase 3 trial. Now we've submitted FDA approval and we've obtained FDA approval for the Pfizer BioNTech vaccine. And that's kind of where we're at now with the Moderna and I think the AstraZeneca as well, is we're very close to approaching that FDA approval kind of stage of the trial.
Right. So like Juan was saying, there's like the preclinical, so in animals. So the vaccines that we're talking about have already been tested in animals. They've already made sure that they're creating antibodies. The immune system is being triggered like we need it to, and that there's no big side effects that we can see. They've already gone to humans, done phase 1, which is they've been escalating, give slightly higher doses until they say what's the safe dose. Then they've gone to phase 2, and then they've gone to phase 3. What's different between this and all the other big vaccines is, I mean, the COVID is stopping the world from functioning like it's supposed to, right? So the American government came up with Operation Warp Speed, which was a joint operation between HHS, Department of Defense, the NIH, a bunch of different governmental organizations to say, do we need this to move quickly, and we need it to happen now, okay? So normally the experiment, doing any trial, any experiment in medicine there's a lot of bureaucratic red tape, right? So it's kind of like, I say the analogy is kind of like, say if you want to get a house built, right? First you have to like go and find the empty land, and you have to like, set up a realtor, then you find the empty land, then you think about how much you want to pay for it, then you try to like bargain from whoever you're buying it from, then you submit an application, you get approved from the bank or whatever, you buy it, then you submit, you create your blueprint, and then you have to submit it to the government. The way that we did this is that there was literally someone standing at every step being like, all right, you're thinking about buying a house. Here's some options. This is how much they're going to cost. We already have the bank loan pre-approved for you. We already have an architect. There's 10 architects that are already designing everything. So everyone's been working on, they haven't actually rushed the trial part itself. The trial is still just as good as any other trial. So it's called, the gold standard in medicine is a placebo-controlled, randomized, double-blind study. And so Pfizer's and Moderna's meet that. They have a big sample size, so 44,000 in the Pfizer group, 30,000 in the Moderna group. The Oxford AstraZeneca is a large number as well in their UK and Brazil trials. So we have a good number of populations. The only difference between this and other big trials is that in order to apply for the FDA emergency use authorization, you only have to wait 2 months from when those 44,000 people have received their vaccine, okay? Normally, you have to wait 1 year for full FDA approval. So the reason why they approve it is because they said, the harm from COVID is so much, we're willing to, and the majority of vaccine major side effects occur within the first 6 weeks. So we waited even a little bit longer than that. We've waited 2 months. We haven't seen any major side effects. And so we think it's safe to go ahead and start rolling. The benefit outweighs the risk, outweighs the harm of going ahead and getting this started. There are still going to be keeping close track to see if there are any long-term side effects, okay? So we don't have the full 1 year like we would for another vaccine because this is an emergent situation. There's an ongoing pandemic. They said 2 months is good enough. That's their rule from beforehand. If there's a pandemic, you need emergency use authorization. You have to watch your patients for at least 2 months. So we have that data on the Pfizer people and on the Moderna people. We're going to continue to accumulate more data. So by the time this vaccine comes out to the general population in say spring or summer, we're already going to have much closer to your full year worth of data. So that's going to continue to accrue. Right now we have 2-month data on all of those patients. The other last thing I'll mention that made things a lot quicker. People here were we're used to working with coronaviruses, right? If this was a novel, a brand new virus that we had never known before, it would have taken longer. But what happened in this situation is people recently had worked for SARS and for MERS. Those are both from the coronavirus family, right? So they'd been working on making a vaccine. The Oxford group, the AstraZeneca vaccine is made by the Oxford group. They have an entire center dedicated to vaccination, okay? January 11th is the date that the people in China said, you know what, this is bad. We need to figure out what to do. So they got the genetic code of that full virus and they uploaded it to the world and said, you know what, open source, figure out what we can do about this. So the Oxford group looked at it on January 11th. they already kind of knew what part of this complex code is going to make a spike protein. The spike protein is what we are targeting knowing coronaviruses. We already know this is the weakness, right? So this is the Achilles heel that we have to go after. So we already knew that. They already had a rough idea what part of that genetic code is going to transcribe that spike protein. So Oxford, they did not know how big this was going to be. Their own researchers say, immediately, as soon as we got the genetic code, we said this might be big. We are going to practice on making a fast vaccine because we know that there might be a pandemic coming in the future. Our job is to make a vaccine. So we're going to work on making it as fast as we can. And they actually didn't have funding when they first started. They were just trying to go for speed. Okay. Later on, when they realized this is growing and growing and growing and becoming more of a problem, they got all of that funding. They had some of the grant money changed so they could make this bigger and start production and all that type of stuff. But that's another reason why you're seeing this everything come out a lot faster than it was before. There's a lot of good reasons. The last thing that I'm going to say, usually when you have a vaccine, first you finish phase 1 and then you do phase 2, and then you finish phase 2 and then you do phase 3, and then you finish phase 3, you start phase 4, then you start production. You find a contract. Okay, this is the factory that's going to make this vaccine. Okay, we're going to get the glass for the vials from here. We're going to get this component from here. We're going to get salt water from here. We had already started manufacturing a lot of different vaccines. So there were over 100 candidates, right? The US government endorsed 16 of them. We'd already started manufacturing them before we had done phase 1, right? Because we said, we're going to put the money in. We just want this to be as fast as is humanly possible. So I think to me, it seems like I don't think that corners are cut where it's important. I think that we've spent a lot of money, invested a lot of money to try to get this out as fast as we can. And I think we've done it in as safe as a way as possible. I think that the trial data that we have, if you had told me 2 months ago even that we were going to have a vaccine that was 95% effective without any major side effects, I would have been over the moon. I would have been overjoyed. I am signing up, inshallah, Friday, 10 a.m. is my dose of the vaccine. So right there is how much I believe that this is going to be safe and effective. The only thing that's left is the long-term potential side effects. There is a small chance of something like that to occur. What did I miss?
Jazakallah khair, Hassan and Muaz, for a brilliant presentation on all of those. Questions have come rolling in, so
Okay.
We can get started on those. The first one is, what are the implications of COVID-19 vaccine, if any, discussed in research studies? I also see Dr. Humaira Ali here as well. She's welcome to chime in.
Dr. Humaira, yeah, please chime in if you hear us say anything, or if you have any things, we'd love to hear from you as well.
Yeah, so this is, what are the implications in research studies? Have you guys read anything like that?
So implications of COVID vaccine in research studies.
Side effects and...
So in terms of side effects, most common, there's side effects are pain at the injection site, fatigue. Some people have had a high fever. Those are the most common things in the Pfizer and Moderna vaccines. Okay, these are different vaccines. So these are both mRNA vaccines. The AstraZeneca vaccine is not an mRNA vaccine. And we'll talk about what that means in just a moment. We can go over that next. The AstraZeneca vaccine and the Johnson vaccine, the Johnson & Johnson trial, both of those trials were halted. One of those, 2 patients had transverse myelitis, inflammation of the spinal cord, which can be pretty serious. Turns out one of those patients had pre-existing multiple sclerosis, which is not diagnosed. No major side effects in the Moderna and in the Pfizer trial. 4 patients had something called Bell's palsy. Bell's palsy is weakness of one side of your face, which can occur from maybe inflammation of a cranial nerve that controls that area. They are not sure if that was directly related to the vaccine because they think that about 4 out of 22,000 of the people that got vaccine, if they just followed a random group of 22,000 people for about half a year, they think that they would see 4 people develop that incidentally. Okay. So in terms of major side effects, luckily for the Pfizer and Moderna vaccine, we've not seen a lot. For the other 2, the trials are still ongoing. So we'll make judgments on them once the trials are completed. AstraZeneca, the UK and Brazil trials, I think, are complete and they seem pretty safe so far. Do you want to address the mRNA topic? Because I think that's a pretty common topic.
As far as what an mRNA vaccine means versus a live vaccine, sorry?
Yeah, I guess.
Yeah, absolutely. So mRNA vaccine, it was like you said, a live vaccine means that you're introducing a weakened virus to the body already. And that is a training exercise. The body will see it, it will identify it. And then, like you said, put a wanted poster up saying, hey, this, whatever this is, this virus, if you ever see it again, we can kind of take care of it. What the mRNA vaccine does, you're not actually introducing any sort of virus as such into the body. What you're doing is you're giving the blueprint to your 3D printer to say, hey, print out the antibodies that will take care of this virus if ever does infect you, right? So all you're doing is giving that blueprint, you're giving that mRNA that's going to make the protein, that's going to make your antibody, and your antibody will take care of the COVID pneumonia, the COVID infection that you'll get if you were to unfortunately catch the SARS-CoV-2 virus.
Yeah, so they found what part of the genetic material of the virus codes for the spike protein. That's the distinguishing fact that we said, hey, we can attack this. The spike protein is on the outside of the virus. It helps the virus get into our cells. So they said, you know what, if we can attack the spike protein, it won't even get into our cells. It cannot replicate without being in our cells. So we may be able to stop the infection very early on, okay? And that was a strategy for the MERS. So they already knew where to find it. So what they've done, the mRNA vaccines that we're hearing about, the Pfizer and Moderna vaccines, they've specifically taken that little bit of genetic code, okay? And they're able to create that genetic code artificially in the lab. They've packaged it in a lipid nanoparticle. All that is like, it's like saying, you know, if you want to send a package to your friend, you put it in a box. So the lipid nanoparticle is just the box that little, the vaccine is sitting in, the mRNA particle is sitting in to keep it safe until it gets from their factory into our system, right? So it's packaged in that lipid nanoparticle. That's what's unstable and requires minus 90 degree freezing for the Pfizer vaccine, or minus 20 degrees for the Moderna vaccine. So that just helps the mRNA, that little bit, get from outside of our body, from their factory, into our system. And then what happens is that mRNA is going to get into our cells. That lipid particle is going to help it get into our cell. And then, just like what the virus does, the virus would just use our cells' machinery to create proteins out of, okay? So what we're doing is, instead of recreating the entire virus, we're creating just that little bit called the spike protein. That spike protein is going to sit on our cells, and our immune system is going to detect it and say, you know what, that's not welcome here. Let's figure out a way to defend against it, so that if we are exposed to the SARS-CoV-2 virus in the future, we already have a plan for what to do. The mRNA vaccine technology, I know it's been controversial. That's been going on for about 20 years, where we've been trying to figure out that we've realized that this is a way that we could do to create vaccines. We've also thought about trying to create anti-cancer medication that way. There have been things that have gotten to clinical trials, but we have not yet successfully created one, okay? This was the time to do it. This is when everyone put all the resources behind it. They said money is not a factor, manpower is not a factor. Do whatever you need, figure out how this technology works, and do it. I think it's pretty brilliant. It has not been, there's nothing in my mind that comes into, like, it does not mess with our DNA. Our DNA sits in a nucleus, okay? The mRNA is never going to enter the nucleus, so it is not interfering with our genetic material. It is using our ribosomes, just like the virus would, to create the spike protein that's going to sit on our cell, and our immune system is going to detect. The AstraZeneca vaccine does not do this. The AstraZeneca vaccine, what they do is they have an inactivated adenovirus, which normally only infects chimpanzees, okay? So they've been able to take this adenovirus and reproduce it in a way that it does not cause any symptoms from us. What they've then done is taken out a little bit of that adenovirus genetic code and substituted it with the COVID, that virus, the SARS-CoV-2 genetic code. So the same thing is going to happen. That virus will infect us. Because that virus is not dangerous to humans, that virus will replicate within us. It will still generate that same spike protein that SARS-CoV-2 would have, and then that our immune system is going to detect that. Similarly, the Russian vaccine, Sputnik V. Sputnik V is actually a human adenovirus, and they've substituted part of that genetic code for the spike protein code, pretty similar to what all the different vaccines are doing, okay?
So are you saying that the Pfizer one is unique because it uses mRNA, but the other ones are more traditional?
Pfizer and Moderna are both unique because they use mRNA as a direct way to enter into our cells and create that spike protein to present to our immune system. Okay. The other two put that genetic code into a pre-existing virus. The Oxford AstraZeneca is.
So is that more a traditional way of doing it?
That's a more traditional way of doing it. But even those are innovative within the traditional ways, right? So it's like, it's modern. It's a modern way. It's not like the old school. The old school way would
Instead of
be,
using the virus, you're using a different virus. So
yeah, you're using a different virus as a vector,
yeah.
as a transportation material.
Got it.
So these are all, I mean, they're all creative ways to do it. And I think it's pretty marvelous that, you know, we've been able to get this far. I think it's a kind of a testament to human advancement and scientific advancement that we were able to do it. Is it new? Yes. Does that mean that it's dangerous? Not necessarily. Are people a little hesitant? Do I understand people's hesitancy to use something new? It's been tried in, you know, over 70,000 people so far. It's going to continue to be tried in more people, so we have a longer time to look at more adverse side effects. I spent the majority of my spring watching people in the ICU die from COVID. Okay, in all seriousness, young people, old people, mothers, fathers. I have seen what COVID does personally with my eyes. And I think at this point, everyone here has family or knows someone who has family who unfortunately has passed from COVID. So the question is not, you know, is this risky? Is driving to the supermarket risky? Yes. Can you have a car accident on the way to the drive to the supermarket? Possibly, right? I'm not comparing the two, but I will say the risk of not being vaccinated and this pandemic going on, you know, continuing on, is immediate, and we know that that exists. So really what we're asking, I think, in my mind, what I'm asking myself is not, is the virus risky? Is the virus riskier? I mean, is the vaccine risky? Is the vaccine riskier than continuing letting the pandemic go unchecked, right? And to me, the answer to that is, give me the virus Friday morning as soon as I can get it. Give me the vaccine Friday morning. So to me, that's kind of what I'm thinking about.
So we do have a lot of questions rolling in. Let's try to spend about a minute per question, if you don't mind. I know it's hard to do sometimes because you want to get into the details. So the first question, Fozia says, how does the vaccine differ fundamentally from other vaccines? I think you've answered that with the mRNA and the other stuff. Is there anything else you would like to add? Someone added a follow-up to that. Why is the COVID vaccine need to be stored in super deep cold temperature, which is different from our own bodies?
Right. So I think that's a very important question. The Pfizer vaccine, minus 90 degrees. The Moderna vaccine, minus 20 degrees. Minus 90 degrees requires super fridges that are available in very few places. So I think in Seattle, it's probably like University of Washington and Swedish have them, and maybe that's about it. The Moderna vaccine, minus 20 degrees, so not nearly as cold. And these are to stabilize the lipid nanoparticle, which is the box that the mRNA vaccine is enclosed in. Because it'll break down if we earlier. That the super cooled is preventing us from being able to distribute as well as we can. Luckily, the Oxford AstraZeneca vaccine, Oxford created this. They've told AstraZeneca you are not allowed to profit from it. The only way we're going to let you have it is you are not allowed to profit from it. This is going to be much cheaper, and you can store it in a normal fridge, just like a polio vaccine and other vaccines. So there's a lot of great hope for us being able to use that in the third world, developing world, to be able to vaccinate people there.
All right, alhamdulillah. Jazakallah khair for explaining all of this. How many doses will be administered for the vaccine to be effective?
So far, the first 3 vaccines that are in the pipeline, Pfizer and Moderna, 2 doses. Pfizer, dose 1, 21 days later, dose 2. It's the same thing that's going to be redosed. Essentially what happens is the first time you generate an immune response, no one knows how long the immune response lasts. No one knows how long it lasts after you get the initial infection. If you were infected, there is an immune response. No one knows how long it lasts. We don't know yet. We've seen people that have been infected twice. We don't know if everyone's immunity wanes or some people's immunity wanes. We don't know if there's mutations and you can be infected twice. So what the second dose is doing is bolstering your immune system, saying, look, we showed it to you the first time. Here's a second look. This is what you want to fight against. So you create more antibodies, etc. So Pfizer, 2 doses. Moderna, 2 doses. Oxford AstraZeneca, 2 doses. Johnson & Johnson, 1 dose. So once again, they're hoping, and there's other candidates down the line that are still being produced. So they're hoping that the 1 dose, you know, if we're going to remote locations in Pakistan and India and other places around the world, it might be hard to follow and keep track of, all right, it's been 21 days since they got their first dose. You might just be able to go there 1 time. So we're hoping for vaccines for those places that are easy to store, cheap to create, and you don't have to dose twice.
Beautiful. One of the differences between this vaccine and other vaccines is that we have not seen enough people or had enough time to see the long-term side effects. One person I know is afraid it could make them infertile. flu vaccine, smallpox vaccine, etc., are tried and tested, whereas this is new. What reassurance do you have to someone who is worried about this?
I would say that traditionally, the majority of vaccines, any major issues have occurred within the first 6 weeks. It's very uncommon for us to have issues later. The second thing that I would say is that the vaccine and this mRNA structure is very simple. It's a very straightforward way of doing it, very utilitarian. So the chances of creating side effects, long-term side effects, are low. That said, Allahu alam. I think that he says with difficulty comes ease. The pandemic has been difficult, and I'm hoping this is his opening his gateway of mercy. If it wasn't for his permission, then we would be unable to create a vaccine without him. So this is my hope and belief. That said, whatever he has in store for us, then we will bear with patience, inshallah.
Jazakallah khair. There's a second question as well about reproductive impacts. So I guess you've addressed that.
I don't know anything about it. Time will tell.
Amal says, I am extremely allergic to penicillin. Would it be okay for me to take the COVID vaccine?
There have been incidences of anaphylactic reactions, which are severe allergic reactions. Those occur. I think there were 2 of them recorded in the UK. I think Moaz was saying one in Alaska. Those are people who have severe immune responses to something. I think time will tell as this rolls out. I think initially they're going to be hesitant to give it to people with severe allergies, just like they're hesitant right now to give it to people who are immunocompromised or pregnant or other things. And as time progresses, I think by the time this rollout becomes bigger, I think we'll find out a little bit more. So I don't think that we know the answer to that yet. I'm hoping. It seems unlikely there'll be a cross reactivity with penicillin, but we'll see as things progress.
Yeah, just to add to that, I think people with severe allergies to vaccines in the past were actually excluded from trials. So there's a little bit of cautiousness approach to it. I think as of right now, the guidance from the CDC and the FDA is if you've had severe allergies, not to penicillin, but to vaccinations in the past, then get it at a place where you can be monitored for a little bit. So I think that's the formal guidance right now.
All right. Does the vaccine protect against transmission of the virus or just prevention of the COVID disease? Can a vaccinated person still transmit the virus and must still wear a mask?
So that's a great question. So the first 2 trials that are coming out, Pfizer and Moderna, what they did is they gave half the people the vaccine and half the people they didn't give the vaccine. They gave saltwater injections, okay? And then the only thing they followed is whether they developed symptoms or not, okay? So the only thing that we know is that there was a lot fewer people who got symptoms in the Pfizer and Moderna groups. We don't know if they were still able to transmit the vaccine or not. I think if we're attacking the spike protein, it seems likely that you're stopping it from replicating, so it's less likely that they're infected, but we do not know that as a fact. My understanding is the AstraZeneca trial, they're actually doing intermittent swabs where they're doing PCRs to actually tell, was this person positive and just didn't develop symptoms, or are we seeing a decrease in the amount of positivity right now? Current guidelines are going to be, even if you're vaccinated, do everything that you're doing currently until we know for sure whether you're actually transmitting it or not. So I don't think we're going to see a decrease in our other precautions at the moment. I'm hoping by the time the next school year rolls around, things are going to be more normal.
Inshallah. Faisal says, were there vaccines for SARS and MERS? If this is the same, why do we need a vaccine in this case?
This is not exactly the same. These are different coronaviruses. So it's similar. We knew that they have a structure, they have spike proteins. But we did not, it's not the exact same spike protein. So it's not going to be the exact same. So the antibody is kind of like a key in a keyhole type phenomenon where it has to be the exact same actual structure for it to be able to open that lock, or in this case, to be able to attack that virus. So because there's not exactly the same, we need a different immune response. And other vaccines, I know they at least got to the late stage of development. I don't know how effective they were, or if the trials were big enough. We had enough patients to enroll that many people to see how effective they were.
And it wasn't as widespread, right? So it wasn't as urgent, it wasn't as necessary, and there wasn't as much funding and so on.
Usually to recruit 44,000 patients, it takes years. There were so many people with COVID that they were able to do it within months. So it's a unique situation.
Yeah. Someone asked, and again, you can also message the questions privately to me if you don't want to message publicly. You can also private message me as well, or Moaz or Dr. Hassan. Um, Someone asks, what about the nanoparticles in the vaccine? People are saying that there's nanoparticles in the vaccine that the government is following and controlling people with.
Uh, I think it seems pretty unlikely. I think the technology required to create nanoparticles. I mean, I don't know. I'm a doctor in a tech city, so maybe the people that are into tech know more about the possibility and capability of introducing nanoparticles. To me, it seems really unlikely because anyone could get a dose of the vaccine and analyze it in a microscope, and if they found nanoparticles, I think that'd be a pretty big controversy. I think it'd be really expensive to do that, and I think the government is tracking us from our cell phones and all these other ways that they track us anyway.
It's too late.
Yeah, I think it's too late unless you're a really smart security guy. You have really good firewalls and VPNs and all that type of stuff set up. Maybe you're safe, but I don't know. It seems unlikely to me. That's the best I can say.
All right. Okay. We do have questions rolling in. We have 13 minutes left, so let's try to get through this. Since memory cells for every pathogen differ, can you address the idea that memory cells against SARS-CoV-2 may be more short-lived than other memory cells? Seems like a technical question.
Moz, what do you think?
I don't think we've proven anything just yet. I think it's impossible to say. And I saw a question that came a little bit later as well, addressing, will we need to be revaccinated down the road? And again, that's something that we don't know just yet. We just don't know that. Could this be something?
If the effectiveness, sorry to interrupt, if the effectiveness is 90%, then that means 10% of the people could use a revaccination. Is that what that means?
So it means that there were two groups in the study trial, right? So in the Pfizer there were 44,000 people, 22,000 people got the vaccine, 22,000 people got salt water. And the 22,000 people that got salt water, I think there was like 170 that had COVID symptoms. In the Pfizer, the vaccination group, only 8 people had symptoms. So 8 out of 176 is like 95%. So not everyone who's going to get the vaccine is going to be able to generate a sufficient immune response. And if exposed to the virus, they may still get the virus in the future. 95% is incredibly high. So the FDA is willing to approve any vaccine that gives you 50% effectiveness. That's the threshold, 50%. So 95% is beyond even what we would have dreamed possible back when all of this started.
So I'm going to jump in here actually and kind of talk about what it actually means to be effective. Effectiveness in these vaccine trials just meant that you didn't develop clinical symptoms. Could you still get the virus? Could you still have the SARS-CoV-2 swimming in your blood? Absolutely. But you could possibly, just not have the COVID-19 symptoms, right? The actual infection itself. So I guess I saw a couple of other questions as well that were asked. If you get the vaccine, could you still transmit the virus? And to answer that question, as far as we know, we're not 100% sure, but as far as we know, we think the answer could be yes. You may still get the virus, but you won't get the actual COVID-19 infection. So I think that's a very important distinction. You can be infected, but you can't. You can still be infective, but you can possibly not have the symptoms. And that's what the 95% effectiveness means, is that 95% of people that got the vaccine did not have symptoms. Did they still get the virus? Maybe. But did they have symptoms? No. So I think that's a very important distinction to kind of keep in our minds going forward.
Yeah, and I think we're going to find out in the near future with serial testing is do you actually get infected or not? My personal guess, if I had to guess, I would say I don't think that people do, but that's completely a guess. They are going to find that data for us and inshallah come up with an answer to that question, because it's a very important question to answer.
Great. All right. Which vaccine is the best to take?
I don't think that we have a choice right now, right? So like for me, I'm getting it Friday. The only vaccine currently approved is Pfizer. Tomorrow, I think Moderna, the Moderna vaccine, I think the FDA is going to meet about that and see if they give it approval. The answer is like, we don't know yet. It seems like the Pfizer and Moderna are 95% effective. Sounds great. The AstraZeneca vaccine has been shown to be about 70% effective. Definitely good enough. Okay. So I think time will tell, if maybe down the road, we're going to see that they are encoding different spike proteins and that down the road, the virus mutates. And then all of a sudden, the spike protein that we were trying to protect against is the virus is gamed it a little bit, and now the spike proteins change, right? So we see that with the influenza, is that the virus changes. It's normal for viruses to change over time, okay? So maybe we'll find out, hey, the Pfizer vaccine was really good early on, and then 8 months down the road, all of a sudden the Pfizer vaccine stopped working, and now everyone needs Moderna. Who knows? Right now, I don't have any information to tell you one is better than the other. Whatever someone offered me, I would take it and say, Bismillah ar-Rahman ar-Rahim.
Why is it that a conventional vaccine wasn't pursued instead of mRNA?
It's faster. Doing the mRNA thing was faster, and we wanted to get it quickly.
Right. Yeah. Conventional vaccines are still being pursued right now. It's just that the Pfizer and Moderna mRNA vaccines just came out much faster due to the nature of mRNA vaccinations.
Yeah, and you're going to see more candidates continue to come up. There's actually a few names for people who are interested in buying stock in companies. There's still a few names. I don't even remember all the names right now. There's actually 16 that the U.S. federal government invested in, right? And that doesn't include the Russian, the China. So there's a bunch of different vaccines that are in the pipeline still. These are just the first comers.
All right, great. We have 9 minutes left, and we have more than 9 questions. So I don't know if you guys have a hard stop. I do have to leave at 9, but I did ask Moaz if he can take over the questions if I need to go. So let's try to get through this. How can we be sure that there will be no double standard in the administration of the vaccine to the Muslim community, considering the discriminatory nature of our country, since minority communities have been medically disadvantaged in the past by vaccination campaigns?
I mean, I think that's a distribution type question. I don't have an answer to that. I know right now, whichever vaccine whoever's being offered a vaccine, it's kind of based on right now. So right now it's healthcare workers and I think nursing homes are the first candidates, because those nursing home clusters are really, really bad. Older people with multiple medical comorbidities, and if it spreads within there, things get really bad. And then it's going to be older citizens and people that are higher risk. So we'll see kind of how things go down the road, but you would hope that there isn't any disadvantage given to minority communities. I don't know who's going to be in charge of making sure that happens.
Mm-hmm. Great. Someone asks, is it the same way the flu vaccine works? And so we learned that it doesn't work the same way as the flu vaccine. So in future, do you think the flu vaccine will cover COVID as well?
No, it's a different virus. They might come up with a combined vaccine, which is like 2-in-1, but most likely they'll do it separately, because if you're allergic to one, they don't want to give you both, and they don't know which one you're allergic to. So you might say annually everyone ends up getting a COVID vaccine and a flu vaccine. Depends on how long the immunity from the COVID lasts, and we don't know the answer to that at all. We're not even close to knowing the answer. The only that we're going to know that is as time passes.
Great. Since we don't know how long the immunity from the vaccine will last, is there a plan to revaccinate the population every 6 months if the immunity from the vaccine is proven to only last for 6 months?
Pfizer would really like that. I think this is going to be people we're monitoring. So the people who have already been in the trials, we're monitoring their antibody levels. They get tested every X amount of days, and that's going to continue for a while. They got their $100 for being in the trial, so they're Pfizer's property now. So they're going to keep testing them. We're going to see how long these antibodies last for. Once the antibodies are gone, the memory cells, all that type of stuff. So that answer is still up in the air. Right now, the focus on stopping the pandemic.
Dalia says, is the fact that these vaccines have been tested under precautions of masking, social distancing, etc., counted into the efficacy results? What are the chances the efficacy will change when people let their guard down?
Yeah, I mean, that's a really good question. You're right that, so these trials were conducted in different countries as well. I think Brazil and Mexico, there's some places. I don't know exactly how much social distancing and all that stuff there was. There definitely was some. So I would assume that if everyone let their guard down, and now people are certainly being exposed to higher doses, then maybe you'd have lower efficacy and a higher infected rate. Totally possible. And like I said, 44,000, an N of 44,000, a sample size in a trial is a good number. Before this hits the general population, that N is going to get way bigger. So we're going to see a lot more people and get more information about that. I would not be surprised if that 95% becomes 90% or 80% down the road. I don't know.
Yeah, again, I mean, just jumping on that question as well. I think it does have a little bit of an effect. Again, just understanding that the efficacy just means that you can get the virus, but the efficacy means that you don't get a serious, serious disease. You don't get a serious infection. So that won't change. When if you get vaccinated, and the efficacy of the vaccine itself will not change, but will we still require us to keep our guard up? Absolutely, yes. We should still be social distancing. We should still be masking. And I think this will last for a while as well. It's still going to be a good amount of time where we'll be living in this quote unquote new normal.
Okay. Jazakallah khair. And when will the vaccine be available for everyone in Washington? Are there any estimates when us normal people will get it?
So it's going to be
Are imams tier 1, tier 2? Where do imams
essential job in my eyes, Sheikh. Frontline worker. Yeah, so it's going to be healthcare workers, and then essential jobs. Essential jobs is going to be, I guess, how Inslee or whatever community they have decides it. My guess is, so Moz was talking about, what was Fauci saying earlier, Moz?
So Fauci, he when this question was posed to him, he very directly says end of February to mid-March. So that's, I guess that's the NIH's kind of belief. I think it's a little optimistic.
Sounds optimistic.
Yeah, it's a little optimistic. I would add maybe a month and a half to 2 months to that.
Will it be quicker for people in cities compared to rural areas?
Ooh, so you know what the way that our strategy seems to be is that we're less invested in the Pfizer vaccine, right? So it's early, but Pfizer actually offered us like 1 million additional doses, and we said no. And we're giving more. I think we're investing more, trying more for the Oxford AstraZeneca, because that one is going to be, like I said, you can just put in a regular old fridge. It's going to be easier to get to rural areas. So I think we're in terms of numbers that we've already pre-purchased. Pfizer is the least, Moderna is higher, and I think Oxford AstraZeneca might be even higher than that. So we're hoping that we can get it out to the rural areas and stuff, but there may be a little discrepancy. And I'm going to be vaccinated. My wife won't be currently. I would not have my hopes up. I don't have my hopes up for her until summer. Okay, summer is where I'm thinking that's probably a realistic thing for me. That vaccine will come down to the general population as well. So, but I'm conservative. I try not to get my hopes up.
Inshallah, before the next school year. I think that would be the best thing.
That's
inshallah, if everyone can get it before. Last question here looks like, is it safe for little kids?
Not included in the experiments. So we don't have an answer to that question.
Okay, no question. Okay. And another repeat of the question, would you infect other people? The answer to that is it's possible. Right. That's what I've heard. All right. Great. So it's been a great session. Jazakumullah khair for the spitfire answers. Alhamdulillah. And for being able to get it done within the time. I really would like to thank both of you, Dr. Aijazi, Dr. Hassan, as well as Dr. Muad, for joining us today. I know we had several other of our community members who are doctors in the audience as well. Dr. Humaira, I saw. So thank you all for joining us and for supporting this. And inshallah, we hope that those that are sick, may Allah subhanahu wa ta'ala cure them. Those that have passed, may Allah subhanahu wa ta'ala have mercy on them and forgive them and give patience to their families. Thank you all for joining us today. We will continue, inshallah, having community conversations on important topics that are important to our community. And we'll be taking a break for the holidays. And then after that, in January, we have 2 presentations lined up on Ivy League colleges and entrepreneurship. So I'm looking forward to both of those conversations. I hope you all will join me. If you would like information on that, you can go to our website, mapsredmond.org, and sign up for the newsletter, inshallah. With that said, that's it from us, and we'll see you all next time.
Oh, can I make a closing remark?
Of course, of course.
All right, I just wanted to say real quick, like I said, I'm not an expert in immunology or vaccination or microbiology. Everything I've done is kind of the best of publicly available information in my research based on that. I apologize in advance for any mistakes. I'm human and I made mistakes, but I'll tell you that we did try our best to give you a good presentation. Any benefit is from Allah, any mistakes and anything that comes bad is from shaitan, from ourselves. That said, I'm a part of the community. I'm around. If you ever see me, you have any questions for me, feel free to get in touch. And I'm your brother.
Jazakumullah khairan. Thank you.
Jazakumullah khair. Jazakumullah khair. Assalamu alaikum wa rahmatullahi wa barakatuh.
Assalamu alaikum.